Alpana, थोड़ा कम हो very. Yeah, that's good. So, good morning, everyone, and I am delighted to see all of you today on the 14th Foundation Day of NIBMG. Truly delighted that all of you have come, even if it is on a Sunday, for this very important day in the life of this institution. This where we have reached is due to the untiring efforts of a huge number of people, and taking each and every name is going to be near impossible. The founding directors, subsequent directors who have held positions, the faculty members who are here today, the fellows, the administrative head, the finance head, the system analysts, and all others who work under them. I would like to thank all of them who have been here today, as of today, but also the ones who have been part of the journey of these 14 years. So these 14 years, we have accomplished a lot. We were a young institute. 2009, we were born, and then Professor Dhrubajyoti Chattopadhyay reminded us that the talk about this institute started about 4 or 5 years before that. We have progressed to be known in India as the premier genomics institute, and we are justly proud of it. We have taken part in multiple national and international projects, and we have done a very good job in that. We have taken part in the call which the nation gave when there was the unprecedented pandemic, and I'm happy to say that we have come out with flying colors. During the COVID, our institute worked day and night on multiple aspects, multiple facets, by which our country of 1.4 billion was able to keep the citizens more or less safe as much as possible. So there is justifiably a lot of things which we have to be proud of and we should be proud of. So having said that, 14 years is a time when in an individual's life, it is probably the best time of the life, when adolescence is almost ending. We are moving towards the early adulthood. And that brings, as you know, each one of you have gone through that time. It brings additional challenges. It brings additional motivation. And it brings additional ways by which we make ourselves a path which will be there for the future. And that is a challenge which NIBMG has to take up as it embarks in the next part of the journey. The challenge to continue to do cutting-edge basic research, but at the same time, not only live up to the concept of basic research, take your basic research to the next possible step. That challenge has to come within us to take the fruits of our discoveries to the masses who are allowing us to pursue our passion. All this is possible because of the taxpayers who are giving us the money. And that is possible, it is we ourselves, and for the students, your parents or your relatives who are actually funding us to take our journey forward, to take our passion forward. It is important at some point we start thinking about how we can actually solve healthcare issues in future using the facilities which we have, using the knowledge which we have, and using the power which we have. Probably many of us don't know what power we have in ourselves. Many of us think that about only today, tomorrow, maximum a month from today. I would want all of you to not think of only today or tomorrow or the next part. Think of the institute as a whole. Come up with plans, come up with ways by which the processes can be smoothened. The institute can work together. Collaborations can be established, not only among the faculties, but also among all the aspects of this institution, so that the process of research for which we are here can go forward. So new areas are going to be taken up in near future. We are trying to augment our student strength. As you know, this year also we have taken significant amount of PhD and IPhD students. We are going to augment our faculty strength, diversify a little bit, so that newer areas come in, so that we get plugged into the mainstream of the biology, at the same time retaining our core expertise. So the core expertise, we are never going to give it up, but going into the biology will be the motto. And that is something which this institute will be passionate about in the next few years of its journey. So this was my overall thought process about it. So I would like to welcome today, before going into the more nitty-gritty of, there are certain things which are lined up, which I think the Sridhar and Saroj will take it forward. But as a welcome address, I welcome all of you to take part wholeheartedly into today's all the things which are lined up, but more importantly also take it part tomorrow. Tomorrow is an open day for our institute. We have actually invited a huge number of students who are going to come from the different schools in the neighborhood and far. So take part tomorrow also. And once they leave, just don't think that this year's Foundation Day is over. Foundation Day is just one day in a 365-day calendar. What is more important is to dedicate yourself towards whatever you are doing for the institute throughout those 365 days. So with this short message, I would like the next part of the program to go forward.
In the past few months, we have organized a few competitions as a part of National Science Day and International Women's Day. And what better day to celebrate the winners than our Foundation Day? So I would invite Dr. Sridhar Chinnaswamy to kindly call the winners of competitions held on National Science Day on stage, and request Dr. Sagar Sen Gupta to please give away the prizes.
Morning everyone. So as you will all remember, we had 28th February, we had the Science Day, and part of that we had two competitions. One was the Science Model Building. So there were about 8 teams that participated, and the first 5 winners were announced. And let me repeat that again. The first 5 winners will have an opportunity to display their models in the Kalyani Book Fair later this year. And the first 3 will also have the chance to go to the IASF Science Festival, which will be sponsored by the institute. So congratulations to the first 5 groups. So let me call out the 5 groups first. I'll call the, I'll give the list of 5 groups, and then I'll call the winners of the first 3 who will get separate prizes. So, the first group obviously is Jyotishman Sharman group. Second was Shupratim Ghosh group. Third was Bhagyashree Pradhanan group. The fourth was Abhishekta and group. The fifth was Arun Kumar Mishra and group. So all these 5 you will be going to the Kalyani Book Fair. So as I said, the first 3 will go to the ISF, and they were also given cash prizes, which we cannot give cash now, so we have converted into gifts, and all the gifts you could see here. So first group, I will call upon Jyotishman Sharma and his team to come over here to receive the gift. So they had a cash prize of 5,000 rupees, so the gifts are of that value. Now the second prize winners were Shupratim Ghosh and group. So can I call upon Shupratim? I think he's on leave or something. So can I ask the next person, Arunima? Yeah. So they had won a cash prize of 3,000 rupees. So that the gifts are of that value. So the third prize was won by Bhagyashree Pradhanan group. Can I request Bhagyashree and his head of group? Yeah. So they had won a cash prize of 1,000 rupees. No problem. 3 is a good number. Congratulations again to all the 3 winners and all the 5 winners who will go to the Kalyani Book Fair. We also had a debate competition, and the topic was artificial intelligence is a threat to human intelligence. So it was a lively discussion. We had for 1.5 or 2 hours time, and we had to announce a winner and a runner. First I'll call upon Jyotishman Sharma, who was the winner. The runner was Deeptanil Vishwas. So there was also an audience round. So about 7 people participated from the audience. So impromptu. So they just walked up and started speaking for or against the debate topic, and the winner from that round was Pritika Quatra. Pritika, I think she was standing outside. Can you guys see her there? Is she there? Okay, she can take it on her behalf. Probably she forgot that she had won this prize. Yeah, there she is. You want to run? Yeah, even I had to remember, and I also. So it was an impromptu. So people just walked in, and the judges didn't know the names. So in fact, I had to find, because the judges had written as Kritika Patra, but she's Pritika Quatra. So I had to confirm the judges again, and they confirmed that it was Pritika. Thank you. Yeah, thank you, sir.
For International Women's Day, as a part of the organizing committee, I'm very happy to announce, call upon the winners. Request Dr. Sengupta to please come forward to give away the prizes. So in extempore, Ms. Dhanashri Ketkar was first, and Mr. Pallov Datta and Kallol Datta was second. In the quiz competitions, we had teams. So team members of the team who stood first was Pratyusha Chikala and Tuneen Ranjan Malik. And team members of the second team were Dr. Molinath Acharya and Jyotishman Sharma. And
You want an action replay? They have forgotten to take it.
Yeah, both of the prizes again actually.
Why?
They are going to leave. This time probably. Yeah.
And finally, the third game that we had was an activity challenge. So there were two teams. The team members of the team that stood first were Devrati Guha Roy, Seema Bhartiya, Anuradha Gautam, Animesh Kumar Singh, Kaveri Shivastav, and Jagashila Das. The team members of the team that stood second were Anjali Gupta, Sukanya Mitra, Jayata Roy, Monalisa Mukherjee, Saheli Pramanik, and Rimpa Nandi. Thank you to all the participants and the winners and Dr. Sengupta. So, today we have the privilege of having Professor Dhrubajyoti Chattopadhyay, Honorable Vice Chancellor, Sister Nivedita University with us today to deliver the Foundation Day lecture. May I please request Dr. Sagar Sengupta, our Director, to kindly introduce our speaker.
So, it is one of the most happiest days of my stay here to welcome Professor Dhrubajyoti Chattopadhyay to my institute in IBMG. Why is it so important? Because the fact that I am standing here today, a huge credit goes to Professor Chattopadhyay. During the time I did masters in Baliganj Science College, Department of Biochemistry. It is the way he showed the path in those years, something like 35 years back, has actually shown me how to go about doing science. So it is a delighted and also an honor and a privilege for me to have Professor Chattopadhyay, I call him Dhrubajyoti, to come over here and give the Foundation Day talk. So I think the name is so familiar, it doesn't really require any real introduction because is a colossus in in the science of West Bengal and India. But still, for the people who are standing here in the back, let me tell a few words about him. After finishing his PhD from the Bose Institute, Calcutta, in the Department of Biochemistry, he joined the Biochemistry Department in the University of Calcutta in 1983. He became the Guha Professor of B.C. Guha Center for Genetic Engineering and Biotechnology under the Department of Biochemistry in 1997. He was the Dean, Faculty of Science at the University of Calcutta in 2003. He became the Director at the Center of Research of Nanoscience and Nanotechnology at the University in 2007. And he was the Pro-Vice Chancellor of University of Calcutta in 2015. After that, he was the founder, Vice Chancellor of Amity University, Kolkata from 2015 onwards. And then he is currently the Honorable Vice Chancellor of Sister Nivedita University in Kolkata. So you understand, from 1983 to 2023, it has been a long journey, and journey not only for science, but for the people who have come in contact with Professor Chattopadhyay. He is a teacher per excellence, a researcher above excellence. He has many papers, book chapters. The fact which all of us, all his students, and there are many of his students sitting here who are the faculty members, directly or maybe indirectly. The most important thing which Dhrubajyoti had was the clarity of thoughts and mind and the ability to put across his viewpoints. And everybody felt that Dhrubajyoti is speaking to him or her at a personalized level. And I don't think I'm talking for myself, whether it is Anupam, whether it is Arindam, it is always has been like that okay, they are thinking that he is speaking to them at a micro femto level, he is understanding their problems. And that is the ability of Dhrubajyoti. He is recipient of numerous awards and distinction, accolades. The most important accolade probably he cares for is the love of his students. Member of BC Goa Research Conference, fellow of all the science academies. So this list goes on. Means if I stand here, it will be I think he is actually in his mind thinking when will Sagar probably stop. Apart from this, one thing which is very important is his love for science. The love for biology and in specifically molecular biology. A researcher par excellence in multiple aspects, and think of the breadth I am going to talk about: virology, molecular virology, transcription, biochemistry, nanotechnology. It shows that how Dhrubajyoti actually has moved with time and how he has enhanced his knowledge and able to light the lives of many others in all these diverse fields. So it is a pleasure, it is an honor again to have him with us on my first foundation day as the distinguished guest. Dhrubada, it's up to you.
Very good morning to all of you. I don't know what's going on inside me, but one thing I can tell you that there are quite a number of my dear students over here. But when students hello, 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 hello. When the students are actually move up and up and up, and they can go to the some place where you are very much, you feel very happy that you could see your student in that place. There are a number of students over here who really made me proud, and this pride is something which is very difficult to explain. The director of the institute, Professor Dr. Sagar Sengupta, I still remember that just I came back in '88 when they are the student actually, that '88 batch. And that particular batch, the students who left for IISc, and how actually I felt like that I used to visit IISc quite often, and whenever I used to go to IISc, there is a get together of my students and maximum number of their batch, but there are number of students from other batches also. And all of us used to have number of meetings, we used to go out for eating, and there are a lot of fun actually at that time. So the thing what you can get it from the students and their achievements, that is actually the best thing what you can get it. So I don't know how to express that, how I felt that when Dr. Sengupta actually requested me to deliver this Foundation Day lecture. Because again, this is a foundation lecture of an institution with which I am very closely associated. It is very, very strongly associated with this from the very concept when actually been developed in 2004. And in 2004, when actually in the government of West Bengal's Writers Building, there was a meeting, and in that meeting 5 persons are present, and Professor Majumdar presented the concept of this institution to the government of West Bengal. So in that meeting actually the whole thing started rolling on. After that there are a number of phases gone, and ultimately with Professor Majumdar achieved this permission to actually set up this institution in 2009. So now in this 14th Foundation Day lecture, when Dr. Sengupta actually requested me to deliver this lecture, it is nothing but, the best gift from me. A student is actually requesting you to deliver the Foundation Day lecture, and that's a gift, a real gift for me. Thank you, Dr. Sengupta. Um, so then I was thinking that what should I tell? I thought my journey after coming back from United States to establish a laboratory in any university, if I can share that particular thing with you, maybe that's going to give you some idea that how to fight for science, for research, and how to be committed to whatever or be passionate about what you are going to do it. I want to dedicate my lecture to my PhD mentor, Professor N.C. Mondol, who actually introduced me with the virology. So he first actually taught us virology, and we got so excited. I particularly got so much excited to do research in virology. So I did my PhD in bacterial viruses, moved on to animal viruses while gone to US. And then while coming back from US, my mentor and a lot of my friends, they said that it's a suicidal decision. Why you are going back to India? You can't do virology back to India. Molecular virology is very difficult to do it in India. So you should not go. And but I thought, let me come back and let me see what I can do. And I was thinking about in virology, which particular thing I should take. A virus which is unknown normally to others, but in India it is been discovered. Let me try to find out any virus like that is there or not. And while searching, I could get from the, that in the university in the Alabama, this particular institution which maintains a viral directories, in their directories there is a name Chandipura virus, and I thought that, oh, this is and then I found that it's a name of a village in Nagpur, and the first discovered in India in 1965. So I thought that let me go back and start working in this field of Chandipura virus, and that particular journey to understand this molecular mechanism of Chandipura virus infection, I shall go through it very quickly, but to give you some idea about what we have done at that time. So if you look at this, isolated 1965, and then later isolated from patient from Madhya Pradesh, from Telangana area, from Gujarat, from Andhra Pradesh, and mainly always we could see the outbreak in the Nagpur area, and in general whenever the outbreak is there, there is a large number of death of kids, children below 5 years, and whenever there is this outbreak, there are number of kids like 15, 20 in that particular area, they actually succumb to death. And then if you look at that time, these particular informations which are the case history which we could get it from the literature, that this particular virus is found to be a major cause of acute encephalitis in children in North Telangana, Andhra Pradesh. But think about this whole area in the map of the India, the area which is being actually this virus is there. No other area you can get this virus. In this particular area you can get the virus. So, and it has been isolated from sand flies in India and West Africa, and the presence of the virus in Africa indicates a wide distribution, although no human cases have been observed on this. Now, after 2010 to 2020, there are a large number of outbreaks, but one particular outbreak is also happened in Uttar Pradesh, and in 2023, there was an outbreak in Gorakhpur area also, where this particular virus has been identified. And in general, the clinical symptoms are just like any other viral symptoms: fever, headache, vomiting, weakness, and you have the neuropathological syndromes. And in general, death within few hours to 48 hours of hospitalization. But the age of the patient is below 5. So it's only below 5 these patients are dying, not above. But in that area, large number of adults are also being infected, but they never actually, immediately they can get rid of this. So in general, this high mortality is observed in human children below the age of 5, but up to the age of 14 to 15, you can see the symptoms. And the virus kills 100% of laboratory mice up to the age of 18 days and 50% of aged mice 19 to 21 days, but older than 21 days do not die. So you can see a parallel between this humana as well as these rodents. And this later on been identified, this is an arbovirus, arthropod-borne virus, and it has got a lot of its friends over here, and ultimately you can see how it comes out with the sandfly. So when the classification been done, it's a mononegaloviridae, that means mono means single-stranded RNA virus, and this is a negative single-stranded RNA virus. It belongs to the Rhabdoviridae, belongs to the family of vesicular virus, and so which has got vesicle stomatitis, Chattopadhyay virus, and it's a very good close friend of the rabies virus, the Lyssa virus. So you can see that there are a list of viruses which have been identified, the one which I have searched before coming over, and this is a list which actually pushed me to work in this Chattopadhyay virus. It's a bullet-shaped enveloped virus, and I have already mentioned about what we could see. Now, this is actually what we could see in the virus. It has got outside the virion particle, you can see that the nucleocapsid protein is there is a lipid bilayer, the glycoprotein, matrix protein, and if you look at this whole RNA, it is encapsulated with this nucleocapsid protein, and there is large proteins associated with it, with the phosphoprotein also. That's actually the viral situation. And if you look at the genome organization, that there is in the genome, there is a leader RNA at the end, and then we have this 5 RNA, and there is the terminator one. So this is actually the RNA structure, and if you look at this viral structure over at the bottom, and so in general, what we could see that during the transcription and then this particular 49 nucleotide leader RNA is being synthesized, but the other RNAs are being translated. Leader RNA never gets translated, and they translated to the proteins of the respective molecular weight, and the large protein is 241 kilodalton protein. And what is actually we wanted to look at in our whole journey is the interaction between this leader RNA and the P protein, leader RNA and the N protein, and then the protein-protein interaction. So these are the things which I am going to share with you. But here I just wanted to mention that one thing, this whole Rhabdoviridae, if you look at the cycle, so after entering, you get the RNA, and from the RNA, you get the transcription. From the transcription, you get the synthesis of the protein molecules, and this synthesis of the protein molecules, you have either the N, P, L, these 3 component together gives a replicase complex, or you can have the virus assembly. The replicase complex replicate the RNA to give the antigenomic template and to give the positive stranded RNA, and this RNA replication goes on. And this transcription, you can see that it doesn't have the N, but it has got the L protein and the P protein in the phosphorylation stage. So this is actually the life cycle of the virus. You know that when I came back to this Calcutta University and I wanted to work in the molecular biology, the first thing what I felt that there is no tissue culture lab. There is in our university, and then also it's very difficult to work with any cell lines and other things. Those things are not there. I was a little bit, what shall I do then? Then I find that, okay, I have I got the virus from the NIV, National Institute of Virology in Pune, and then isolated the RNA, and then try to find out that whether I can actually do some sort of cloning of the genes of this, and clone the genes. The first gene is the smallest gene I have chosen, is a P gene, and then I cloned that, and then again I cannot actually express it into any other tissues, any other animal cell line or anywhere. So I have to express it in the bacterial cell line, and I expressed it into the bacterial cell line. here I can tell you that when I presented this after coming over here in presence of the DST, Professor Padmanabhan was there and Professor Jyotirmoy Das from IICB was there. They are in the committee members. They said, animal virus you are going to do express in the bacteria, is that going to work at all? But I don't have anything in my, I have to express in bacteria. I said I shall try and see that what I can do. You know, that's the thing which actually helped me. Because nobody else, because whatever molecular virology, everybody actually worked with the cell lines or the animal cell expressed this. I don't have that particular thing, so I expressed it in bacteria. So I got a particular P protein molecule, and that P protein molecule, when I actually tried to have the transcriptase complex, this transcripted complex, you can see the PL, this particular L protein, which is a bigger protein, which I can purify from this complex, and then we have the P protein, but this protein is in unphosphorylated form. And that never helped in the transcription. And if that never helps in the transcription, the first step actually I cannot do. So there is no way I can do the transcription. So naturally, this P protein in unphosphorylated form is the first thing which helped me to do some research. Because nobody else actually looked at. Everybody is looking at the P protein, the phosphorylated P protein. So I started with the P protein unphosphorylated form and then tried to do that whether this P protein I can actually get it phosphorylated and what particular kinase can actually phosphorylate it. So I got this animal cell culture from IICB and then purified the cell extract, and using that cell extract trying to do the phosphorylation of this P protein. The first thing I started is whether I can do the phosphorylation of P protein. I could get the phosphorylation of P protein. So I can just mention to you. So the first thing actually, my student Dr. Debashish Chattopadhyay, who is in the NIPGR, what actually he did, this P protein, cloned it and then characterized that this P protein is actually the transcriptional activator, and this particular P protein, what we synthesize is unphosphorylated P protein, it can be phosphorylated by casein kinase 2, and the casein kinase 2 is phosphorylating at by site-directed mutagenesis and other. I have shown, my student have shown that it is a serine 62, a single site phosphorylation is actually there, serine 62 phosphorylation. So the cloning and the single serine phosphorylation within the acidic domain of Chattopadhyay virus, P protein has been shown by him. And when we actually did this particular P protein with the CK2, which phosphorylates the P protein, and it actually in the serine residue, we used that particular phosphoprotein and then we did the transcriptional activity assay. And what we can see that this transcription is possible only when it gets phosphorylated and phosphorylated at the serine 62 position. And when this serine 62 position being identified, then another student actually started to prove that if phosphorylation is not there by site-directed mutagenesis, changing this serine 62, and then also looked at the effect of phosphatase on the phosphorylated P protein, and then looked at how it worked out. And when they have looked it out that whether it helps the transcription or not, and it is possible to show that this phosphorylation induced a major structural change in the N-terminal domain of the P protein of Chattopadhyay virus. Now, if I cannot get this unphosphorylated protein, I cannot do these experiments because I don't have anything. That's why I got this unphosphorylated protein, and I can demonstrated this, and then actually demonstrated that this N-terminal region of P protein is responsible for phosphorylation mediated homodimerization. Now, throughout this vesicular virus world, people are looking for how this replication, the same replicase and transcriptase, and how the replicase behaves as replicase, and when it actually acts as a transcriptase. Nobody knows about it, that how this transition occurs. So I thought that let me try to resolve this issue. So what I have shown that with this particular protein, the regular biophysical work, the phosphorylation induces some sort of conformational change in the P protein, and when this conformational change is happening, then only it is actually helps in the phosphor transcription. So Dr. Shobin Boshak, who is also in National Institute of Immunology right now, he actually demonstrated that this particular, we have the P0 complex, we have the phosphorylated protein complex, we have the mutant P complex, and then we actually did all the transcription, and this transcription activity, what we have demonstrated, it is possible for us to reconstitute this transcription in this particular virus molecule itself, and in the in vitro transcription assay with the detergent disrupted virus we have shown, and it's also shown the expression profile of the phosphorylation status of wild type and mutant P protein in transfected Viro76 cells. Now, what we actually at this time we have our own tissue culture system. So, in our university we set up our cell culture system, we started our cell cultures and other things. So, we actually started infecting the cells with the different things, and it is possible for us that it is P protein only in the phosphorylation form what is actually going to do. The viral titer goes completely down if the P protein, the Senin62 is going to be mutated. So, the experiment looks like this, that the effect of the P wild type or the P mutant protein on virus yield. And this is by the transfection assay, we infected with the virus and then also transfected with the P clone or the P mutant clone, and then look at the virus yield. And what we can actually demonstrate that with the RT-PCR assay, it is possible to demonstrate that whether, if you remember that either RNA means discrete small molecules, and when we said replication, then there is no transcripts, but there is a genome. So, either these junction regions are going to be in the PCR product, that means it's replication, and if the junction regions are not there, these are actually transcription. So, in that way, we try to actually look at the level of the transcription and the replication in this type of assay. So, one is to look at the virus yield, another is to look at the transcription and replication. And when we actually looked at this unphosphorylated P protein and demonstrated that this particular P protein participates in replication. Now, this cannot be possible if I cannot have the unphosphorylated P protein. So, the replication is actually where unphosphorylated P protein participates, but in transcription the phosphorylated P protein participated. That could be demonstrated, and then it is possible for us to say that why this is happening. There is some sort of leader RNA, if you remember at the end of this leader RNA and the interaction of the leader RNA with the others is very important. So, when the phosphorylated protein makes a complex, then we get the transcriptase, and when the unphosphorylated protein makes the complex, it gives a replicase. So, this is the first time it's being demonstrated that in this type of negative stranded mononuclear virus rhabdoviridae, it is a unphosphorylated protein which is being, that means right after infection the newly transcribed RNA molecule gives rise to unphosphorylated P protein. That unphosphorylated P protein gets replication, and because of this actually it is possible for us. So, now once we publish these particular papers and try to convince people that this may be the switch between the transcription replication, then the Encyclopedia of Virology, they are actually contacted, and they said that why don't you write the chapter on Chandipura virus in the encyclopedia and also mention about this mechanism. So, this is how actually it is possible for us to go for this mechanism. The phosphoprotein P in its unphosphorylated form interacts reversibly with leader RNA, and this reversibility we actually could demonstrate by this, and also the specificity of the binding, RNA binding could be demonstrated. Now, while doing this, it is possible for us that phosphorylated P does not interact with leader RNA, but there is a distinct involvement of two separate hubs of the leader RNA. If you remember, leader RNA is a 49 nucleotide, and in the leader RNA there are two different separate hubs. Either it can form complex 1 or in the form of complex 2. And unphosphorylated phosphoprotein P specifically interacts with leader RNA, whereas phosphorylated totally abolishes this interaction. So these complexes could not be formed. And if the complexes could not be formed, that is actually going to give us this particular information that leader RNA binding ability of virus P protein is regulated by its phosphorylation status. There is a monomer and dimer of the Cheripura virus unphosphorylated P protein which binds leader RNA differently, and that has got an impact on the RNA synthesis itself. Now, then Shubhrudeep actually, who is still now in United States, he actually demonstrated and evaluated the structure of this P protein, and it is possible for us to evaluate the structure of the P protein and find out that in this structure a negative stranded RNA virus probing intratryptophan energy transfer, he used that particular methodology to find out the single and the tryptophan mutants of this P protein also, and demonstrated it. Then Dr. Aruna Roy, who is still in the United States, he started to work in the status of this leader RNA expression in the infected cell, how actually this thing is there. And so he tried to find out the intracellular expression of the leader RNA, and this intracellular expression is actually coincides with the viral replication. And I mentioned to you that we developed this particular assay, which we call as the semi-quantitative RT-PCR assay, to measure the level of replication, to measure the level of transcription. And thereby it is possible for us to make a real-time analysis of this viral replication and transcription. And it is possible for us to demonstrate that the virus immediately after infection goes for transcription. After some time, when you have no unphosphorylated, all the phosphorylated P protein is being exhausted, some unphosphorylated P protein started to accumulate in the system, the virus complex goes to the replicase mode, and you can see the replication started. And this actually gives us a real-time analysis of the viral replication and transcription. And it is also possible for us to demonstrate that this expression of the leader RNA, it also coincides with the viral replication. So if you remember, I mentioned about this particular structure, and then we showed at what particular level the leader RNA is expressed within the viral infected cells. And it was found that this is actually abundant 6 hours post-infection. So that actually clearly tells us that when actually the transcriptase is going to be changed into the replicase form. And then we wanted to know that does the P protein interact with the leader RNA in the viral infected cells. We have demonstrated it in outside the cell, the in vitro experiments. Now we want to demonstrate it in vivo one, and it is possible for us to do the RIP, the RNA immunoprecipitation against the P protein, and demonstrate that how actually the expression of P protein happens within the viral infected cells. And it is possible for us also to demonstrate that phosphorylated P does not bind to the leader RNA within the viral infected cells. And that actually clearly supports what we are looking at. Why it could not actually do this? So that also again we have proven by the demonstrating the structural changes in the unphosphorylated P protein during the leader RNA binding, and how this particular tryptophan mutants actually not to organize itself in binding it. So the role of the leader RNA in switching the polymerase from transcription to replication is this, that right when the virus infects the cell, what happens? It has got all the phosphorylated protein along with the L protein, and this particular complex then starts moving on, and while it is moving on, it behaves like a transcriptase complex. It actually starts doing the transcription, but during replication what happens? This P0 is actually binding, and the leader RNA transcript guides the polymerase to continue on the genome itself. So this is how actually get we either transcription or the replication. Now then we did some peptidomimetic approach for actually going for an antiviral development, And when we looked at it and tried to look at the alignment of the P proteins of different rhabdoviruses in the C-terminal conserved domain, and it is possible using the NMR studies by doing the chemical shift perturbation with the synthetic peptide corresponding to the C-terminal domain of the P with the leader RNA. And that actually demonstrated us that if we use this synthetic 36-mer peptide, and you know that to get the peptide inside the system, we have the 6D arginine at the end, and then use that particular thing to look for either that particular peptide or a mutant peptide. And all these things we used for our understanding how it actually alters the viral replication. So the in vitro conformation of this particular binding we have studied, and then the ex vivo effects of this peptide on the viral infected cells we studied, and we could get that this viral infection actually reduced with respect to this particular this mutant peptide. So wild type peptide, there is reduction in IC50, but with the mutant peptide of that, it is found that it is not going to, it remains at the same stage. So the wild type peptide shows significant antiviral property, and while by viral titer assay as well as by measuring this, it is possible to demonstrate that. And then with this particular peptide, we did that same semi-RT-PCR, and then with that, it is possible for us to look at the replication or transcription inhibition of the potential of this wild type as well as a mutant synthetic peptide. And it was found that the wild type peptide specifically inhibits viral replication, but not transcription. So replication, that means the first stage of this particular virus after going into it, it's transcription. But after transcription, we get replication. First stage is not going to be affected with this peptide, but it's a second stage where the viral production, progeny production is going to be there, then that is going to be completely affected by this wild type peptide. And then if you remember, I mentioned to you the other protein is the nucleocapsid protein and the P protein. This interaction also we started to study about it, and Amitabh, who is also right now in UK, he actually demonstrated by cloning and demonstrated the different structures of this N protein, and then this he showed that the P protein is actually, it mediated chaperone activity on the N protein. So by the effect of osmolytes and chaperone-like action of P protein and on the folding of the nucleocapsid protein been shown, and also it's possible for us to show that the P protein is an N protein specific chaperone. It's not a chaperone in general, but it's chaperone only, it's N protein specific chaperone, and it gives the N protein in its proper form. So what type of chaperonic form is there? And parallelly, Dr. Raja Bhattacharya, who is right now in Amity University, he actually studied to look at the N protein interaction with the leader RNA and demonstrated that it forms an RNase resistant complex, encapsulation complex, because you know that the very start of application in the encapsulation, and the encapsulation is been shown by us, and this N protein either in its aggregated form or in its single form, monomer form, we can actually look at it, and N protein forms a soluble aggregate, and the aggregate N cannot form nucleoprotein complex, and that is going to be shown that how it is failing in doing the LRNA leader RNA encapsulation process, because at the very beginning when the leader RNA is there at the end, the replicase complex shifts over there, and the replication complex has got the N protein, and the N protein, if it is in the aggregated form, it cannot do this. So this has been shown by this experiment. And then the phosphoprotein provides specificity of the RNA binding ability of the N protein. So N protein's RNA binding ability is also demonstrated, and it is possible for us to show that N protein forms two different complexes with leader RNA, and these two different complexes, one complex is actually important. So the model so far is like that: N protein, NP complex formation. So the specific recognition of leader RNA, and this is what we call as a nucleation complex. And once this nucleation complex is formed, it starts encapsulating the RNA molecule. So encapsulation of the RNA molecule is very important, and if the non-specific aggregation of there, if P protein is not there, then N protein goes non-specific aggregation. Non-specific aggregation of N protein recognizes any RNA sequence, but specific one only forms the nucleation complex. So this has actually been demonstrated how this is important for the chaperone activity. And then we did the functional domain mapping of N to show the specific areas, which particular domain is important for formation of which specific complex. And while doing that, Dr. Arindam Mondal, who is right now in IIT Kharagpur, he actually showed by protease digestion assay the structural organization of the N protein and how this structural organization is going to be very important for this encapsulation molecule. So while looking at this TEM picture, it is possible to show that how actually this N protein, if you remember that complex which is being formed, and this complex is a bilobe morphology, it has got a bilobe morphology. And this particular morphology, then the mutant N proteins are being synthesized, and then this has been used for the self-association, and it is demonstrated that oligomerization status of the mutants are actually also demonstrated, and then it is demonstrated that what is the specific region which actually helps in the oligomerization of the N protein mutants. Why it is important? That the N protein encapsulation is the first step in this particular viral replication process. And it was found the mutant N1320 have similar ring-shaped morphology like the wild-type protein, and they can form this type of encapsulation complex under the electron microscopic view. So there are two domains which play crucial role in the oligomerization process. One we call as an initiatory domain, and there is another we call as a modulator or the helper domain, which helps in the self-association, and this self-association occurs with the proper structure formation is not possible, and this is actually how we have to look at it in the case of the starting of the aggregated complex, which has been first demonstrated by Raja Bhattacharya. So RNA interacting domain has been characterized by Arindam Mondal, and it has been found that this RNA interacting domain either in the monomer form or the oligomer form, and the monomer N recognizes leaders specifically, whereas the oligomer N enwraps any RNA non-specifically. So this is actually what happens in the actual life cycle, and when this oligomerization is there, then it is going to bind with any RNA molecule, and that has been shown by different type of things. And so in that respect, it is possible for us to demonstrate that with respect to RNA interacting domains also, we have the two separate domains in the N protein. One particular part enwraps the whole RNA genome non-specifically, and another is the initial specific recognition by the C-terminal region. So this is most important in the viral life cycle, where this recognition by the C-terminal region is actually becoming very important. Now, these are actually the facts which helps in the, so this is actually to tell you, my students, that there are only 5 proteins, and these 5 proteins has to do so many different types of activities to keep itself much ahead of the cell itself, which has got so many different genes. So that itself, by doing differently, that which particular part interacting with other particular part, how it actually helped the things to done. And so we came to this conclusion. So we elucidate the functional domains of virus nucleocapsid protein, which is involved in the oligomerization and RNA binding, and this is actually help us to get the encapsulation complex. Encapsulation complex means when it actually having the replication. So all these things actually demonstrated that our understanding about the interaction of the N protein with the leader region, how it actually gives rise to the formation of the encapsulation complex. The non-specific one is not encapsulation. The specific one is the encapsulation. And if there is encapsulation, then it becomes RNase resistant, and non-specific one, when there is no encapsulation, then actually it is RNase sensitive. And this NP interaction then we wanted to look at in the within the cell, how it happens, and you can see that then this is actually the formation of these particular things. What we can see with different ratios of N and P, we start demonstrating this binding of the N and P, and it is possible for us that what is the stoichiometry of number of N protein and the P protein required for this interaction. If you remember, I mentioned to you that it's a monomer binding protein and the dimer binding proteins are there. So we have two different ratios used, and then we actually shown that how actually the two interact under the two different situations. So one situation is that each of the NP protein are actually going to this, and the other one is the in this situation. And this NP forms, so two different complexes. One is a oligomer NP complex, another is what we call as a monomer NP complex. And then this NP interaction, we use the different N mutant proteins and the other proteins over there, and looked at that self-association domain is there, oligomerization domain is there, and if you remove any one of the domain, how it actually work out. And then with the his tag assay, it is possible for us to demonstrate that this particular, where actually we can get the actual interaction. And it has been demonstrated that this is actually the region where the actual association gets there, and these particular points are very important for the N molecule itself also. And then we do the, in general, the association assay by immunoblotting, by co-immunoprecipitation with anti-P, and demonstrated that which particular area is going to be very, very important. And it was found that this 1 to 320, which retains the structure in the electron microscopy, the N-terminal P interacting site, and P exert the chaperone-like activity upon that area. So the chaperone-like activity is going to be in that area, and we did the co-transfection assay, and from the co-transfection assay, the chaperone activity also inside the cell could be demonstrated. And then we wanted to know that can this P interact with oligomeric N in vivo, and it is possible for us that P interacts with monomeric N only, but fails to interact with oligomeric N in ex vivo condition. So this is how actually we can say that the P protein is inhibition of the self-aggregation and the non-specific RNA binding, while the nucleocapsid-like structure formation and non-specific RNA enwrapping has been done by the oligomeric N protein. And this is actually how we can recognize the NP protein interaction. So we have a little bit of a development of this then Arunabha also wanted to know about, at that particular time, people are looking at the virus as a some sort of factory within the cell is there for the viruses, and how actually we can look forward to that. And by using the different antibodies and different tag proteins, so we want to know that where the RNA synthesis occurs. And while doing that, it is possible for us that CHPV inclusions, which contains the N, P and L proteins, and in CHPV infected cells, the CHPV P, L protein co-localized with the N protein in the viral inclusions. And these inclusions are actually demonstrated by this, and you can see the merging also is over there. And so encapsulated genome with the polymerase complex, this is a one what we can actually demonstrate also inside the cell by this experimentation. And then we looked at the viral inclusive activities. We call it the morphogenesis of the VI particles during the viral infection, and we demonstrated that how this is actually happening during the different infection process. And if we do the parallel with it with the viral replication and transcription, it is possible for us also to demonstrate that what happens after 2 hours post infection, 4 hours post infection, 6 hours post infection, and the 8 hours post infection. So in general, these viral inclusions, aggregosomes, viral factories, these are there, and we know that different types of viruses has got different mechanisms of this. And over here, the informations are not available for the negative stranded RNA viruses, but it's possible for us to demonstrate in negative stranded RNA viruses, it is the viral inclusions which plays important role. And we compared that, and to prove that it is not the comparison with the aggregosomes, and it is possible for us to demonstrate that these are not aggregosomes, but these are actually the viral inclusions which are formed. And this comparison with aggregosomes, again it is possible for us to demonstrate that these viral inclusions, it contains other than this, the HSP70 and the ubiquitinated proteins, and this is actually the one which shows us that it is viral inclusion, it is not the viral aggregosomes which is formed in this case. And we did the FISH assay with the viral RNA specific probes, and by doing that it is possible to demonstrate that how this non-specific or the role of microtubules also in the morphogenesis of these viral inclusions. And it is possible for you know that using the different types of inhibitor complexes, it is possible to demonstrate, and we have demonstrated what happens after 4 hour post infection, what happens after 8 hour post infections. And in this particular case, we have shown that how these viral inclusion bodies are being formed, and this role of microtubules in morphogenesis of these viral inclusions. So this is a very interesting observation, that involvement of the microtubules also in forming and keeping the viral inclusions inside the system as a separate body inside the after infection. And this role of microtubules in morphogenesis of these viral inclusions also we have studied. We also tried to do an in vivo nascent RNA labeling with the bromoiodine labeled UTP, and it is possible for us to show that what happens with this in presence and absence of NOC, and in vivo nascent RNA labeling with this actually giving us a very interesting observation about these inclusions. You can see these inclusions are there, and how this inclusion merges when this we could get it over here. And there is actually the one which demonstrates this viral inclusion, microtubules involvement, and everything. Now, effect of microtubule disruption on the virus when we try to study, it is possible to demonstrate that the effect of microtubule disruption on viral transcription is there, and effect of microtubule disruption on viral protein production also we have studied. So what we can say right now that viral factorials, which are the probable model. So the virus after coming over here, the RNA is going there, and you can see that they are actually viral inclusions. In viral inclusions, the virus starts actually synthesizing these RNA molecules. And lastly, what I want to say about the last student's work, who actually wanted. So we now know about the factory. We now know about the replicates, we now know about this particular thing, but still no idea about how the virus goes inside the cell actually. Is anybody? To identify the cellular factors that are responsible for CHPV infection, and to elucidate the mechanism by which the identified host proteins propagate the CHPV infection. So we actually did these experimentations by using the co-localization of the N protein and the L protein in the CHPV cell, and at different times after infection, and looked upon these particular co-localized things. And then we isolated the viral inclusion bodies and studied them in the cell-free system with antibody against the N or L, or by merging with that, and it is possible for us to demonstrate the presence of the viral genomic RNA and viral transcript in inclusion bodies. Now, interestingly, what it is possible for us to demonstrate that N, P, L, and M viral proteins, they are found in the cytoplasmic pellet in the 6 hours post-infection, but the absence of the nuclear fraction from this CP is also very evident. Now, when we actually add the EBP1 is another host protein, and gamma-actin, these are detected in the CP in 20% fraction as well as the CP 0%. Now, unconjugated ubiquitin is largely absent from both fractions, and the conjugated ubiquitin might be present. What does that mean? So, when we demonstrate this and the role of EBP1, this is very interesting to demonstrate, but because you know that recently the RSV virus infection, it has been demonstrated, EBP1 plays very, very important role. So, the fluorescent staining of this virus to visualize the CHPV receptor interaction, and we try to actually identify that how actually it will work. After this, we did this treatment, TRITC, strain viruses on the cells, and by double infection, it is possible for identifying this. But what interesting thing, what we could see that in presence of the alpha-2 microglobulin, what happens, and in absence of that, what happens, and then it could be demonstrated very easily that these are the cases where the host protein factors are getting involved in the viral infection in a very crucial fashion. And this then we try to validate with the FRET, and we do the FRET validation of the CHPV with the A2M, another protein interaction, and then it is possible for us that how this particular CHPV A2M actually interacted. And A2M presence increases the, so you take the cells, transfect with A2M DNA, you can see more A2M receptor, and you can see the viral infection is also getting higher. And so A2M increases the binding of the virus with host cell, and consequently increases the infectivity of the virus, indicating a positive role of A2M in CHPV infection. So this is the first time a receptor which helps in the viral infection also been identified, and it is possible for us. You know, the A2M has different functions in the cell. So there are so many different functions in the cell, and then it is just like the ACE, what we could see in the COVID, the A2M plays important role in this negative stranded viral infections, and it is possible for us to localize this with the LRP1 receptors and with the GRP78 receptor, but the interaction with the A2M receptors are all dependent on the LRP1, but not with the GRP78. And thereby it was found that this activated A2M complex with CHPV for binding to A2M receptors. Now we know that amantadine, it interferes with A2M binding to its receptors, and it also reduces CHPV infection, which again supports the observation that A2M plays very important role in the CHPV infection, and it is essential for CHPV entry via the A2M receptors, and there is quite a bit of reduction takes place if we take this LRP. Though A2M is there, if LRP is removed, you get less infection. So LRP helps the A2M to be in the correct format to get the CHPV to inside the cell, and it is right now we know that the different mammals, birds, fish, invertebrates for these different A2M receptors which are actually important, and it is demonstrated that the A2M receptor is going fine. So here I just wanted to say that this particular paper with the A2M receptor is just being accepted 3 days back, and so we are expecting that ultimately we can at least go to that format of the receptor site also. So that gives us some sort of satisfaction for me, because I started with unphosphorylated P protein. Thank you everybody. These are my things, and these are the people who helped me to do all the research work. So this is something which they are the really the pioneers who worked so hard for getting so much work during my last many years. Thank you.
Thank you, Professor Chattopadhyay, for that, for sharing your journey and how you converted challenging situations into opportunities. May I now request our director, Dr. Sagar Sen Gupta, to kindly felicitate Professor Chattopadhyay.
Wonderful. This is from our NIBMG.
This picture is about
We have a history behind it. It is the Down syndrome.
oh, okay, all right. Thank you. That's a tradition. Thank you very much.
So with this, we now come to a close for this session while we'll continue our introductions with Professor Chattopadhyay for the rest of the day. We appreciate for everyone's joining us today, and if everyone could kindly get together outside the model area for a group photograph, please. Thank you.
Take care of this.